Okay, students, I thought I would take this opportunity to lecture very quickly about the 20 amino acids. This is going to be a very important part of the course, so it's best that you do memorize the structures of all 20 amino acids. However, for the exam, I'll have you use your flashcards, so it's very important that you get the flashcards of your amino acids done. We have 20 amino acids that the body uses to make into proteins. So I thought I would go over all 20 amino acids along with their corresponding structure. So amino acids are actually the building blocks of proteins. So the proteins that are in our cell are actually a combination of 20 amino acids. So it's not like you have to memorize 20 different structures. All amino acids have a general backbone. So let us begin here with our title, which we will call the 20 amino acids. Put that in here. So all amino acids have a general structure. They will have an alpha carbon. They will have an alpha amino group. This will have a positive formal charge. They will have an alpha carboxylic acid group, and then they will have what we will call an R group. Okay. So the 20 amino acids really differentiate in their characteristics by this R group. Okay, so this R group can be polar, it could be nonpolar, it could be hydrophobic. That really defines the characteristics, the identity, so to speak, of an amino acid. So once again, we have an alpha carbon. Okay. This alpha carbon is usually chiral, except for the amino acid glycine. Glycine is the most simplest amino acid. It's non-chiral, but all the other amino acids will be chiral. That means that this carbon will have 4 different substituents attached to it. So we have the alpha carbon of the amino acid, we have the alpha hydrogen of the amino acid, we have an alpha carboxylic acid of the amino acid, and then we have the alpha amino group of the amino acid. So sometimes this is called the alpha amino group. This is called, since it is attached to the alpha carbon, the alpha carboxylic acid group. Okay. Down here is the alpha hydrogen. All of these substituents are attached to the alpha carbon. And of course we have our main functional group, that R group that really defines and determines the identity of the 20 amino acids. So we'll just call this the R group. Now before we go into the specifics of all 20 amino acids in terms of their structure, there is something I'd like to point out to you, and that has to do with this concept of a pKa. So don't get bogged down with this. We will have another lecture on pKa's. But really for now you should realize that a pKa is just a proton that could come off. So usually this alpha amino group has 1 proton that can come off, and that has a pKa, and that pKa, again, generically speaking, is about 9. 9 for the alpha amino group of an alpha amino acid. Okay. Likewise, the alpha carboxylic group of an alpha amino acid generally has a pKa of around 2. Okay, so it depends on the specific amino acid that you're talking about. Right, so we have not really discussed about pKa's formally, but pretty soon in later lectures you will actually utilize pKa's to determine the isoelectric point of an amino acid, the isoelectric point of a protein. But for now, a pKa just tells you how well a proton is ionizable. So one of these protons can come off of the alpha amino group, and it usually comes off at around pKa 9 or a pH of 9. And one of the protons, well, this one proton of the alpha carboxylic acid group of an alpha amino acid usually comes off at around 2. Now notice the approximations. Okay, so a specific amino acid will have a specific pKa. And then some R groups will also have an ionizable proton. Not all, but some of these 20 amino acids in this catalog will have an ionizable proton, so that means it will also have its own pKa. Okay? So there's a lot really to digest here. Before we talk about proteins and the enormous complexity they have in the human body, we really have to talk about the building blocks. The building blocks are amino acids. A typical amino acid will have an alpha carbon, attached to it an alpha carboxylic acid, whose lone proton, usually has a pKa of 2. Once again, that means that at a pH of 2, the proton comes off. Attached to that same alpha carbon is an alpha amino group, which has one of its protons come off at roughly around pH of 9. Okay? And then we have an alpha hydrogen. This alpha hydrogen is not ionizable. I've seen many students say that this alpha hydrogen has a pKa. Well, it does not. Okay, so there's no pKa associated with the alpha hydrogen of an alpha amino acid. And then some R groups also have their own pKa. All right. So this is the basic skeleton of an amino acid. One little complexity that I want to turn here and talk about right now is that usually biochemistry is talking about stuff that's happening in the cell. Okay, this is a course that deals with the biology or the chemistry of what happens in the cell. So if we're going to talk about the cell, we really have to talk about the pH of a cell. So there is a concept or a designation for physiologic pH. Let me put that up here. And we'll just call that 7.6 for now. Okay, so I've seen some textbooks call that 7.5, 7.3. It varies. Okay, it also depends on the type of cell you're talking about, whether it's a red blood cell or a brain cell or hepatocyte. So if we're going to call a typical generic cell having a typical generic pH of 7.6, what happens to the amino acid is that at 7.6, which is what we will call or designate the typical pH of a cell, at 7.6, this alpha amino group, that proton still stays on. But at a physiologic pH of 7.6, what happens? Well, at 7.6, it's far greater than the pKa of this proton, which comes off at around 2. So actually what happens is that this proton comes off, if we're talking about the physiologic pH or a typical pH inside the cell. So we have to actually modify this if we're going to talk about a typical amino acid in a typical condition of the cell. So we will modify that by talking about an alpha amino acid at physiologic pH. So an alpha amino acid at physiologic pH would actually look like this. Okay, everything is the same. Okay, the alpha amino group, one of its protons comes off roughly at pH 9. Okay. We have the alpha hydrogen, which really is a nice stable bond. That's not ionizable. We have the R group. We don't know about this R group yet, but I'm going to go over all 20 of these R groups and hence all 20 of these amino acids with you. And now the alpha carboxylic acid of the alpha amino acid, actually with a pKa of roughly around 2, that proton comes off. Okay? So at physiologic pH, we don't have this, but if we're going to talk about cellular conditions, we actually have that. Okay? So this is what we would expect a skeleton of an amino acid to look like at physiologic pH, which we designated 7.6. It can also be 7.5. Some textbooks call it 7.4. Again, it really depends on the type of cell. So really, if we're talking about inside the cellular soup, inside the cytoplasm, this is what the proper structure is of an amino acid. Again, simply because the pKa of that proton is roughly 2, and the pH of our cell is 7.6. Okay, that proton comes off at 7.6. Okay. All right, now let's go into all 20 of these amino acids, step by step. Remember, use your flashcards. Memorize these as best as you can. It's going to be very important later on. but for the test you can use your flashcards. All right. So let me zoom back in here, and let's just get started. We have 20 of these to knock down, but again, we really just have to memorize our R groups. All right. So let's start off very simply here. All right. Now I'm going to draw the fully protonated form of the amino acid. All right. So here's our first amino acid. Here's our alpha amino group. Here's our alpha carbon, the alpha hydrogen. Here's the alpha carboxylic acid. And our first R group is very simple. Okay? That is an H. All right. And this is the amino acid called glycine. It is the simplest amino acid. It has a 3-letter abbreviation of GLY, and it has a 1-letter abbreviation of G. 1-letter abbreviation of G. Okay? Now, a couple of things about glycine. Obviously, it's a very simple amino acid, but as a result of this side chain being hydrogen, this carbon loses its chirality. So glycine is the only amino acid where the alpha carbon, or the only alpha amino acid, where the alpha carbon is not chiral. Okay, all the other amino acids in our body are chiral, which means they could be in the D form or the L form. Our body, for whatever reason, uses the D form of the amino acid. All right. So glycine, the very first amino acid. All right, this is going to be a non-polar side chain. In fact, all the amino acids that I will be talking to you about are non-polar side chains. So we'll talk about category 1, non-polar side chains of the amino acid. All right. So glycine is number 1. Let me go up a little bit here. Let me just figure out the space issue here. Well, why don't we do this here? Okay, we have glycine. Let's talk about our second amino acid here. The second amino acid has a side chain with the methyl group. Methyl group. And that amino acid is called alanine. 3-letter abbreviation ALA, 1-letter abbreviation is A. Okay. Now there's one thing I forgot, and that's very important, so I'm going to have to move back here, so you have to excuse me for a moment. Okay, glycine is an alpha amino acid, the simplest alpha amino acid, but I forgot something very important, and I forgot to mention the pKa's. All right, so you should know now that the pKa's of the alpha amino group are going to be around 9. So for glycine, I have the exact value. The pKa for glycine in the alpha amino group is going to be roughly around 9.78. Okay? And then the pKa for this proton, so one of the protons will come off at a pH of 9.78, start to come off. And then this proton of the alpha carboxylic acid group has a pKa of 2.35. All right? All right, so remember, all the way back here, alpha amino group roughly 9, alpha carboxylic acid group roughly 2. All right? So for glycine, it's 9.78 for the alpha amino group, and for the alpha carboxylic acid group, it's going to be 2.35. Okay, 2.35. So we actually have specific values for those. So nonpolar side chain glycine. Alanine, okay, has a side chain that is the methyl group, still nonpolar, and it will have a pKa for the alpha amino group. Can you guess? Well, it's going to be roughly about 9. So the pKa for that one is going to be about 9.87. And on your flashcards, don't forget to write the pKa's for the alpha amino, alpha carboxylic acid, and in some cases the side chain. And the pKa for the alpha carboxylic acid group, this proton, it starts to come off at a pH of about 2.35. All right. So alanine, one letter abbreviation is A. All right, moving on. Our third amino acid is going to be valine. Again, here is our backbone, alpha amino group, alpha hydrogen. I'll draw the alpha hydrogen on top. Here's the alpha carboxylic acid, protonated. The side chain for valine actually is a little bit more complicated. It's a CH with a CH3 and a CH3. Okay, so that is the side chain for valine. Okay, valine. 3-letter abbreviation VAL, 1-letter abbreviation V. Okay, 1-letter abbreviation V. Again, same, nonpolar side chain. You would expect valine, or a protein that has a lot of valines, to be embedded inside the protein structure, because this is considered a hydrophobic side chain. It would want to get away from water, where water is everywhere in the cytoplasm or in the cell. Okay, so valine. There's a lot of clinical consequences for valine. Sickle cell anemia results from a mutation in the hemoglobin protein where you have valine, and then this hydrophobic pocket likes to clump with other hydrophobic pockets. So the valine essentially aggregates, the hemoglobin aggregates because of an amino acid substitution. So that's the etiology behind one variant of sickle cell anemia. So anyways, valine. Let's talk about the pKa's. Okay, 2.29 for the alpha carboxylic acid group, that proton, and 9.74 for the alpha amino group, one of the protons in the alpha amino group. Next amino acid on our list will be leucine. Okay, so we have a long side chain on leucine. So again, as before, our alpha amino group, alpha proton, alpha carboxylic acid. Now, what is the side chain on leucine? It's going to be a CH2 and then a CH, and then our 2 CH3s. Okay. So here is leucine, the R group of leucine. It has a 3-letter abbreviation of LEU and a 1-letter abbreviation of L. Okay, L. Okay. pKa's of the alpha amino group for leucine. We have one at 2.33 for the alpha carboxylic acid proton. And then for one of the protons in the alpha amino group, we have a pKa of about 9.74. Okay, so put those pKa's in your flashcards. All right. It'll be important when we go over some problems later on in the course. All right, still we're at the nonpolar hydrophobic side chain. So these 4 amino acids, glycine, alanine, valine, okay, glycine is all the way up here. Okay, all of these have side chains that are going to be hydrophobic. They will be nonpolar. Okay, so we would expect these amino acids to sort of bury themselves into the interior of a protein. Right. So let's move on to our next amino acid on our list. Okay. The 20 amino acids continued. and we're still on category 1, which is nonpolar side chains. Let's talk about isoleucine. Previously, we talked about leucine. Now let's talk about isoleucine. So here is isoleucine, alpha amino group that's protonated. Here is our alpha carbon with the alpha hydrogen. Here is our alpha carboxylic acid. I'm going to keep that protonated. And then the side chain of isoleucine is going to look a little bit complicated here. CH3. Okay, so that carbon has an H attached to it. It has a CH3 attached to it. And then we have a CH2 and a CH3. So this is a variant of leucine. It's an isomer of leucine, and it's called isoleucine. All right, so here's our side chain once again. And this is isoleucine. Okay. Now the 3-letter abbreviation for isoleucine kind of varies. But actually in your textbook they call it ILE, so that's fine, we'll go with that. The 1-letter abbreviation is I. pKa of isoleucine, 2.32 for the alpha carboxylic acid. And then here for the alpha amino group it's going to be 9.76. Okay, again notice roughly 9 for the alpha amino group, roughly 2 for the alpha carboxylic acid group. By the way, what would isoleucine look like in the cell with a physiologic pH of 7.6 or so? Okay, do you know the answer to that? A little bit of a diversion, but okay. At pH 7.6, if we designate that as the physiologic pH, okay, at 9.76 this proton still stays on. But at 7.6, yeah, this proton is probably fully off. So it actually, isoleucine is going to look like this. Okay, the alpha amino group. Here's the alpha proton. Okay, it becomes an alpha carboxylate. An alpha carboxylate. Here's the alpha carboxylic acid version of isoleucine. This is the alpha carboxylate version at pH 7.6. And there's really nothing to do with the side chains. Okay, there's no ionizable protons in the side chains. Okay, it's still isoleucine. All right, it's still isoleucine. So this is a confusion point for many students. Okay, this pKa just basically tells you at what pH that ionizable proton comes off. Okay, this is still isoleucine. Notice the proton is gone now at physiologic pH 7.6. It actually starts to come off at a pH of 2.3 and above, that proton. All right, continuing on, again, realize we're talking about a nonpolar hydrophobic side chain. Finished with isoleucine, let's continue to move on. We still have a ways to go. But we've actually made a little bit of stride, because now let's talk about our second category of amino acid side chains. And I'll call this nonpolar side chains. We're still at nonpolar side chains, so we'll just stay with that. A couple of more nonpolar side chains of amino acids before we move on to the side chains that are hydrophobic and acidic and basic. All right. So the next one is actually going to be methionine. So I got a little bit confused because methionine actually is an amino acid that has a sulfur group in it, but it's still considered a nonpolar side chain. So here's the structure of methionine. This is the fully protonated form of the backbone, And here we go, methionine, CH2, CH2S, CH3, okay? That is methionine. This amino acid is called methionine. It has a 3-letter abbreviation, MET. The 1-letter abbreviation is M. Okay, here is our side chain. Okay, it's still a non-polar hydrophobic side chain. The alpha amino group has a pKa of methionine. For methionine, it's going to be roughly 9. Let me give you the exact number. It's 9.28. And then the alpha carboxylic acid group of methionine has a pKa of about 2.13. Okay, again, notice it's roughly around 2 and roughly around 9. Okay, so methionine, non-polar amino acid side chain. We would expect methionine to be buried, or its side chain to be buried inside the protein's primary structure. All right, the next amino acid is kind of weird and wonky, so let's move on this way. Our next amino acid on our list is actually a cyclic amino acid. And sometimes in the literature they don't call it an amino acid, they call it an amino acid. Okay. An amino acid, because it's a cyclic. The molecule has undergone an intramolecular cyclization. So this is the amino acid proline. So let's talk about proline. Here's the structure of proline. Okay, so we have the alpha H, the alpha hydrogen. We have the alpha carboxylic acid. And then the molecule is actually, as I said before, turned into a ring structure. This was formerly the alpha amino group, that nitrogen. But the molecule has sort of turned itself into a ring. Okay? And this is called proline. 3-letter abbreviation PRO, 1-letter abbreviation P. Okay, you're going to have to know these 1-letter amino acid abbreviations. There's no way around it. Okay, P is proline. Okay, so one thing is about proline, there is really no. Well, this is still considered an alpha carbon, but the molecule is actually cyclized. It's turned in on itself in an intramolecular cyclization. So we still have an alpha carbon. So this carbon is still alpha. It's still chiral. We still have pKa. So let me give you the pKa's of the alpha carboxylic acid group of proline. 1.95. Okay, notice here it's a little bit lower than our generic 2. The alpha amino group of the cyclized amino acid proline has a pKa of about 10.64. Okay, again a little bit over our average of 9 and 2. Okay, so proline is a cyclized amino acid. One thing that is very important to know about proline, and let me just tell you that right now, bends and turns in the protein's 3-dimensional structure. Okay, so when we start thinking about these entities, these amino acids, and we build on them to make these complicated protein molecules, we have to incorporate their 3-dimensional structure. And anytime you have a proline in the protein's amino acid sequence, if you see a P in the protein sequence, you can try to assume or predict a bend or a turn in that protein structure. Okay, so it's kind of a theme involving proline. Okay, a proline molecule, when it's in an amino acid sequence, tend to cause the protein to turn or bend in that three-dimensional structure. Okay, so proline is associated with bends and turns. It's the only cyclic amino acid that's found. It's actually called an amino acid formed from an intramolecular cyclization. All right, proline is P. Another thing about proline, it's the most conformationally restricted amino acid. So we'll talk about that when we talk about Ramachandran plots. Conformationally restricted amino acid. The most conformationally restricted amino acid. Okay, what do I mean by most conformationally restricted amino acid? Not a lot of degrees of motion. Okay, bonds tend to rotate. It's hard to rotate and have bond movement and bond spinning and turning when you're kind of confined in a ring structure. So it's an amino acid that sort of restricted in terms of its degrees of freedom in terms of bond rotation. And again, simply because of the nature of the ring structure. So it's the most conformationally restricted amino acid. We use that when we talk about Ramachandran plots. So this is foreshadowing future lectures in the course. Okay, Ramachandran plots, which kind of tells you where the amino acids like to be in terms of their structures. And how much degrees of freedom, how much bond rotation, how much bond translation, and all of those types of movements the molecule has. Proline, because it's a ring structure, doesn't have a lot of that. All right, moving on. Proline. Now let's go to our next amino acid. All right. We've got 20 of these to go, so bear with us. Make your flashcards and memorize these structures and know their one-letter amino acid abbreviations. All right, NH3, alpha proton, alpha carboxylic acid group. What side chain will this be? Okay, it's going to be this side chain, a CH2 followed by a benzene ring. Okay. This is a famous amino acid. It's called phenylalanine. It has a 3-letter abbreviation of PHE and a 1-letter abbreviation of F. Okay, you see P has already been taken by proline. So F is the 1-letter amino acid abbreviation for phenylalanine. Let me quote to you the alpha amino group pKa. pKa for the alpha amino group is going to be around 9.31. pKa for the alpha carboxylic acid group should be about 2, and according to the textbook, 2.20. All right, phenylalanine. Okay, here is the side chain of that. Okay, notice now we're coming back to our original structure of an amino acid. Alpha amino, alpha carboxylic acid, side chain. We kind of hit a detour with proline. Okay, so no real side chain in proline. Okay, it's kind of one of those weird amino acids. No real side chain in proline. Okay, back to normalcy, I guess, with our 20 amino acids. So phenylalanine, okay, a very big, bulky side chain. Okay, if you ever look at or build a structure of phenylalanine using your organic model sets, you'll find out that this is a big side chain. Okay, so that's going to really influence its degrees of freedom. That's going to really influence if it's in a protein, where it resides in the protein. It's going to reside in the interior, but it could clash with other side chains of other amino acids just by nature of it being so big and bulky and taking up space. All right, so be aware of that when we start thinking about joining these amino acids together and what type of structural fold, what type of three-dimensional structure they would like to adopt. Okay, so phenylalanine. One clinical disease related to phenylalanine. It is more involved with a defect in phenylalanine metabolism, and that is phenylketonuria, which I think some of you may know or may not know, but we talk about that a lot in our Biochemistry 2 course. It's a disease where the body cannot further metabolize phenylalanine. Because the body cannot further metabolize phenylalanine builds up. The body panics, it doesn't know what to do, it converts it to phenylketones, another byproduct. It incorporates and accumulates in the urine, and a lot of bad, nasty side effects associated with phenylketonuria. But anyways, that is a result of aberrant amino acid metabolism. And for now, phenylalanine is just a very important amino acid. All of them are a very important amino acids for our proteins. Again, sidechain probably would govern that to be on the interior of the protein, away from the outside, away from water. All right, moving along, let's go to our next amino acid in our list. We're almost there in our stretch. Okay. 20 amino acids continued. Let's just zoom out of here. Maybe. Let's zoom back in. Right, we're still on category 1, nonpolar side chains. Our next amino acid that will also have a nonpolar side chain is going to be tryptophan. So, like all amino acids, alpha amino group, alpha proton, alpha carboxylic acid. And now, tryptophan. Its side chain is a little bit complicated. It has a CH2 group. It has this ring structure here. Okay. So here's the side chain. And this side chain actually has a name. It's called the indole functional group. All right. pKa's alpha amino group for tryptophan is going to be 9.41. The alpha carboxylic acid group of tryptophan will equal 2.46. 2.46, and this is tryptophan. Okay, TRP is its 3-letter amino acid abbreviation. 1-letter amino acid abbreviation, W. Okay, so phenylalanine is F, tryptophan is W. All right, be aware of these 1-letter amino acid abbreviations. They'll come back to haunt you. So there's only one thing you can do, and just memorize those 1-letter amino acid abbreviations, because they are not direct. They are not direct. Okay, they just require memorization. All right. Finally, we go to category 2 of our amino acid side chains. Okay, all along we've been at category 1. Category 2, we're going to call them the uncharged polar side chains. Uncharged polar side chains. Okay. So, moving along, let's talk about, so these are the side chains that are going to be polar as opposed to non-polar, which we talked about before. So What would we expect for these side chains if they're polar? They would interact with water. They will interact with water. So if we're thinking about a protein, a more complicated combination of these 20 amino acids, these amino acids would tend to stick out and be associated with the outside of the protein, be associated with the outside milieu, the outside environment, the outside cytoplasm where water is abundant. All right. So by virtue of their side chains, let's talk about our first amino acid with a polar side chain. Everything is the same as before, except our side chain now is CH2OH, and that is the amino acid serine. Okay, here's our side chain. Okay, notice we have a potential for hydrogen bonding with that hydroxyl group as part of the side chain. All right, oxygen is a very electronegative element. It would like to associate with water, which is on the outside of the protein in the cytoplasm. So pKa's, the alpha amino group of serine will come off, that proton will come off at around 9.21. The alpha carboxylic acid comes off at around 2.19. Okay, so 2 and 9. Okay, pretty close to our generic values we've established for ourselves. This is the amino acid serine. It has a 3-letter abbreviation SCR, 1-letter amino acid abbreviation S. 1-letter amino acid abbreviation S. All right. Next one on our list of a polar side chain, NH3. Okay, what will our side chain look like for this? It's going to be C with an H, a CH3, and then an OH. All right, here's our side chain. Again, that polarity comes from that OH group. And this is the amino acid threonine. A 3-letter amino acid abbreviation for threonine is THR, and 1-letter amino acid abbreviation is T. All right. pKa's for the alpha amino group of threonine would be about 9.10. For the alpha amino group, that proton will start or begin to come off at a pH of around 2.09. Okay, so that has a pKa of about 2.09. So in the cell, physiologic pH, this would remain as NH3+, and at a physiologic pH of the cell at 7.6, this will be COO-. The proton comes off at pH 7.6. Okay, threonine. All right, next on our list of nonpolar side chains, let me turn this over. All right. Alpha amino group, alpha proton, COOH. All right, a little bit of a complicated side chain here. No ionizable pKa for the side chain. All right, and here's our side chain. Asparagine. Okay, it's very tricky. Okay, polar side chain, the NH2 interacts with water. Okay, very polar. Nitrogen very electronegative. C double bond O, that oxygen will interact with water. So proteins that have asparagine, we would expect them to be sort of situated outside of the cell, maybe on the outside of the protein, or we would expect them to be where there's a lot of other polar side chains involved. All right, so pKa 8.72 for one of the protons in the amino group, pKa of 2.14 for that carboxylic acid proton. All right, so no third pKa. All right, our next amino acids, we're on category 2, polar side chains. It's going to be the amino acid glutamine, NH3+. You're beginning to see a trend here. Okay, we're beginning to see a trend here, hopefully. Here is glutamine, CH2, CH2, C double bond O, and H2. Glutamine. Here is our side chain of glutamine. Okay, very polar side chain by virtue of the nitrogen and the oxygen. Let's do some pKa's here. pKa of glutamine is going to be 2.17 for the alpha carboxylic acid group. The pKa for the alpha amino group is going to be roughly around 9.13. This is the amino acid glutamine. One of the more important amino acids with regards to nitrogen metabolism within the cell. Okay, glutamine, urea, the urea cycle, getting rid of excess nitrogen waste, toxic nitrogen waste. Okay, glutamine is a central hub for nitrogen metabolism, besides being an important constituent of proteins. Glutamine, three-letter amino acid abbreviation GLN. All right, here we go. One-letter amino acid abbreviation Q. Q. Okay, some people like to call this glutamine Q-amine. When they try to memorize the one-letter amino acid abbreviation as Q. Q-amine, glutamine. One-letter amino acid abbreviation Q. All right. Polar side chain amino acids. Right. Still there, halfway to go, but we're getting there. All right. So. All right. Category 2. Polar side chain amino acids, or amino acids with polar side chains. All right, next up on our list is tyrosine. What does the side chain of tyrosine look like? It's CH2, a benzene ring, and an OH group. Okay, that is tyrosine. Here's your side chain of tyrosine. So we have a big bulky aromatic, and then we have a polar hydroxyl group. So probably the hydroxyl group would want to be with the water or interact with other polar substituents. The big bulky aromatic would want to probably interact with other hydrophobic partners. All right. So this amino acid is called tyrosine. And TYR is its three-letter amino acid abbreviation. What is its one-letter amino acid abbreviation? Y. Okay, Y. I can already tell you there will be lots of problems if you don't memorize those one-letter amino acid abbreviations. Okay, so it's just something you have to do. Okay, if you want to get into these higher level sciences, biomedical programs, medical school, they want you to know these one-letter amino acids abbreviations. All right, pKa of the alpha amino group, what would you expect that to be around? You would expect that to be around 9, so 9.21. What would you expect the pKa for the alpha carboxylic acid group to be around? You would expect that to be around 2. And the more accurate number is 2.20 for tyrosine. Okay. Now, finally, we have a ionizable proton in the side chain. This proton has a pKa. So if we call this pKa 1, that comes off at around 2.20 pH. pKa 2, that comes off at around a pH of 9.21. We have a pKa 3, which comes off at a pH of its pKa, and the pKa of this proton is 10.46. Okay. So we kind of hit a little bit of a bump, because now we have seen an amino acid that has a third pKa, a third ionizable proton. So this proton starts coming off at its pKa, which means at a pH of 2.2 and above, it starts getting removed. One of the protons of the alpha amino group begins to come off at a pH of 9.21 and above. And then as we reach a pH of 10.46, this proton on the side chain of tyrosine begins to come off at pH 10.46 and above. All right, so this is when it begins to ionize at a pKa of 10.46. Okay. So I'm using pH and pKa kind of interchangeably. All right, but just realize that when your pH exceeds your pKa, the proton begins to come off. That's all I'm trying to tell you. In fact, that's where the definition of pKa originates from. All right. Okay. pH is minus the log of the hydrogen ion concentration. pKa is minus the log of the acid dissociation constant. Okay, remember Gen Chem 2. All right. All right, tyrosine, a third ionizable proton with its side chain. This is its structure. Tyrosine. Our next amino acid on our list, NH3+, COOH. It's going to be the amino acid cysteine, CH2SH. Here is our side chain of cysteine. It has a 3-letter amino acid abbreviation of CYS, a 1-letter amino acid abbreviation of C. This is a thiol group, THIOL, SH is a thiol group. And let's talk about pKa's. The alpha amino group would probably come off at what? Probably around 9. This one actually comes off at around, let me look at my notes here. It actually comes off at not 9, but 10.70. Okay, so a little bit of a anomalous amino acid. Okay, it's a little bit higher than our average of 10. And a pKa for our alpha carboxylic acid one comes off at a little bit lower, 1.92. And then we have a third pKa for the side chain, and that's going to be around 8.32 for the SH group. Okay. So 3 pKa's for the amino acid cysteine. And the most important thing I have to tell you about cysteine, get this embedded into your heads now, students. Okay, cysteine likes to form disulfide bonds. Disulfide bonds, okay? One of the most powerful features of this amino acid is that if you have a cysteine in the amino acid sequence with another cysteine away from it, maybe 2 or 3 or 10 or 100 amino acids away from it, they have the potential of those cysteines to join together and form a disulfide bond. Okay, a very, very powerful force, a very, very powerful covalent bond, considered very strong, and it's a very powerful proponent of protein structure, okay, when we start thinking about these in 3-dimensional space, disulfide bonds. So cysteine forms a disulfide bond with itself. The 2 SH groups join. So let me draw for you that structure of a disulfide bond when 2 cysteines join with one another. Okay. So first I'll draw cysteine 1. Here's its side chain. And here is the beginning of our second cysteine. And now here is cysteine 2. And this is a disulfide bond. Okay, disulfide bond. a disulfide bond has occurred between this cysteine and then the cysteine below it. All right? So if there's two cysteines adjacent to one another, or maybe several amino acids away from one another, they can form a disulfide bond. Okay? It's a powerful tertiary structure determinant. Okay, more on this later. A very powerful tertiary structure determinant. All right, so cysteine, 3 ionizable protons, 1-letter amino acid abbreviation C, and it can form a disulfide bonds. For now, when you think of cysteine, just think of the potential to form disulfide bonds. All right, very important to know. Okay. NH3, CH, COOH. And now we are going to go into a side chain that is category 3, which is the basic side chains amino acid. Okay, amino acids with a basic side chain. So category 1, what was category 1? Nonpolar. Category 2 of the side chains was polar, uncharged. And category 3 is what we're going to talk about now, basic side chain amino acids. Okay. So the first one on our list is the amino acid lysine. Okay, it's a very long side chain. 4 methylene groups, 4 CH2s, and then stuck in there is the reason for its basicity, that NH3 group. All right, so here's the side chain of lysine, a basic amino acid. Let me just write that right properly. Lysine, 3-letter amino acid abbreviation is LYS, 1-letter amino acid abbreviation is K. K. Okay, not straightforward. Going to have to memorize K as being associated with lysine. Basic amino acid, pKa of the alpha amino group of lysine should be around 9, and actually it is 9.06. The alpha carboxylic acid group is going to be around 2. The actual number is 2.16. And then we have a third pKa at pKa3, and that is for the epsilon nitrogen. This is called the epsilon nitrogen. And that one has a pKa of 10.54. All right, so 3 pKa's for lysine. So now we had some amino acids that have shown to have an ionizable proton in their side chain. Okay, so be cognizant of that. Be very aware of that. All right. So in your flashcards, make sure you put those pKa's in, not just of the alpha amino and the alpha carboxylic group, but also of any potential side chains. We see it with lysine. We saw it with cysteine. We saw it with tyrosine. All right. Our second amino acid that has a basic side chain is going to be arginine. So lysine and arginine are your basic amino acids. Side chain of arginine is going to be a CH2, a CH2, a CH2, and then we have an NH. Then we have a C attached to another NH2, and then this C has a double bond with another NH2, or with an NH. Okay. Actually, it's an NH2. All right. So here is the side chain for arginine. This actually has a name. It's called the guanidium side chain. Let's talk about pKa's. pKa for the alpha amino group for arginine is going to be 8.99. The pKa for the alpha carboxylic acid group is going to be 1.82. And then we have, because it's a basic amino acid, because it's a basic amino acid, we will also have a ionizable proton for the side chain of the guanidium group of arginine, and that one comes off at 12.48. All right, so 3 pKa's for the amino acid arginine. So notice, just like with lysine, let's write this down here. Arginine. Okay. 3-letter amino acid abbreviation ARG. 1-letter amino acid abbreviation. Can you guess what it is? Okay, it's not very common, or it's not straightforward. It's not direct. Okay, it's just, you got to memorize it as R. So notice the basic amino acids have a side chain whose proton comes off at a basic pH. So arginine, okay, will have a proton that comes off at a basic pH of 12.48. And then going back up to lysine, it has a side chain proton. One of the protons will come off at the basic pH of 10.54. All right. So 2 basic amino acids. Okay. One more basic amino acid. So, well, looks like I can't seem to draw histidine, so there's an N here. There's an NH here. All right. So here's the side chain of histidine. So I have another double bond here. So very important that you get these side chains right in your flashcards. All right, histidine, HIS, 1-letter amino acid abbreviation of S. pKa of histidine, we have the alpha amino group that is going to be at around 9.33. Alpha carboxylic acid group of histidine is going to be about 1.80. And then we have a third proton. This proton, the third proton, pKa of 6.04. Okay, that is the side chain of the imidazole group that has an ionizable proton. All right, so this is the amino acid histidine. Okay, 3 pKa's. Okay, one is for the common alpha carboxylic acid group, one is for the common alpha amino group, and then we have a third pKa at 6.0 for that proton on the side chain. So this would be pKa1, this would be pKa2, and then this would be pKa3. Okay, the amino acid histidine. All right, 2 more amino acids and then we are finished with all 20 amino acids. Okay? And the last 2 amino acids belong to the 4th category of amino acid side chain, so we'll call this category 4. And these are amino acids with an acidic side chain. Okay, so we just went through the amino acids that have a basic side chain. Okay, that's going to be lysine, arginine, and histidine. And now we'll talk about 2 amino acids that have an acidic side chain. So we would expect a 3rd pKa, whereby that proton comes off at an acidic pH. All right. So here we go, NH3 plus. Here's our alpha proton, alpha carboxylic acid group. Okay, I'm drawing it in the fully protonated form here. So CH2COOH. Okay, this is the carboxylic acid functional group. Do we remember from organic chemistry what a carboxylic acid functional group is? Okay, that's the carboxylic acid, COOH. All right. So this is the amino acid known as aspartic acid. Okay. It has a 3-letter amino acid abbreviation of ASP, a 1-letter amino acid abbreviation of D. Okay, D. Okay, again, not very intuitive. 3 pKa's. pKa for the alpha amino group is going to be at around 9.90. pKa for the alpha carboxylic acid group is going to be at around 1.90. And then the pKa for this proton, we have a 3rd pKa, and that's going to be at around 3.90. Okay, so 3 pKa's. We have a pKa1. We have a pKa2, 3.90, and then we have a pKa3. Okay. By the way, okay, we have aspartic acid. Okay, so don't get panicked if you see the structure that looks like this. What's the difference between this structure here and this structure on the left? If I zoom out. Okay, the difference is that the proton has come off. So obviously this is at a higher pH. And sometimes this is called aspartate. Aspartate. It's still D. The thing is that the proton has come off. Aspartate. Kind of like acetic acid and acetate. Okay, conjugate acid, conjugate base pairs. Okay, this would be the acid, this would be its conjugate base. This one, proton, comes off above 1.90 at its pH. This proton comes off at a pH above 3.90. All right, so aspartic acid and aspartate. All right, I've seen many students confuse the two. No, okay, think of acetic acid, acetate. Think about that conjugate acid-base pairs that you've learned in general chemistry too. Right, the final acetic amino acid that we will discuss is glutamic acid. Okay, the only difference between glutamic acid, which I drew now, and this one is the addition of a CH2 group. Right, here is our side chain. By the way, here's our side chain for the above aspartic acid. This is called glutamic acid. Three-letter amino acid abbreviation GLU, 1-letter amino acid abbreviation E. E, okay? E. You just have to memorize it. Just like glutamic acid is an E, aspartic acid is a D. There is no rhyme or reason. That's just how it's going to have to be. All right? pKa for the alpha amino group for glutamic acid is going to be roughly around our conventional 9. It's actually 9.47. And then the pKa for the alpha carboxylic acid group is going to be 2.10. So that's kind of in line with our number of 2, because we said that this comes off at roughly 2. And then this one comes off at a pKa, or this one has a pKa of 4.07. Okay, so 3 ionizable protons for glutamic acid. 1 is for the alpha carboxylic acid group, 1 is for the alpha amino group, and 1 is for this side chain proton. Once again, okay, we have glutamic acid, and then we have its conjugate, which is glutamate. Okay, this is called glutamate. Now, in this version of glutamate, I've drawn this proton has coming off, or has deprotonated. So obviously the pH that I drew this structure has to be above 2.10. And then I drew the side chain proton to come off and be deprotonated. So obviously the structure at which I drew this has to have a pH above 4.07. Okay, so whenever the pH is above your pKa, that's when this proton starts to come off. And I've kept the proton on the alpha amino group at NH3+, that usually starts to come off at a pH of 9.47. Physiologic pH 7.6, 7.5, that alpha amino group stays the same. So those are our 20 amino acids. I just want to give you one final thing before I depart. Okay, don't forget, okay, if we're going to talk about physiologic pH, whether it's 7.6, 7.5, or 7.4, depends on what textbook you're using. Okay, the alpha amino group stays protonated. The alpha proton doesn't do anything. This alpha carboxylic acid group comes off, that proton comes off at a pH of 7.6. Okay, pH within the cell. If we designate 7.6 as the pH within any generic cell, that proton is going to come off. Why does it come off? Because it has a pKa of roughly around 2. Okay, so do not confuse pH and pKa. Okay, the pKa tells you at what pH that proton begins to come off. Okay, it really tells you the acidic nature of that protein. Okay, pKa is derived from Ka, the ionization dissociation constant for a specific proton. All right, so when your pH is above 2, that means it has crossed the pKa of this proton and it starts to come off. All right, and then at physiologic pH of 7.6, well, we have to really look and determine what amino acid we're talking about. Does it have an ionizable proton? Okay. If it's aspartic acid, yes. If it's glutamic acid, yes. If it's tyrosine, yes. If it's leucine, yes. Okay. If it's glycine or alanine or threonine, no. Okay. So just be aware, these amino acids, their structures are really, really dependent on what pH you're going to draw the structure. Okay. I cannot emphasize this enough. Okay. It really depends on what pH you're talking about. If it's a very basic pH, all of these protons will come off. If it's a very acidic pH, we would expect all these protons to remain on unless we bypassed the pKa of that proton. All right, so a very, very crucial point, a very important point. It's a point that students very easily to stumble upon, and we're going to emphasize more about this during lecture. All right, thank you, and we'll see you in lecture. These are our 20 amino acids. Put them in flashcards, use them on the exams. Remember the one-letter amino acid abbreviations, and the faster you memorize these, the better it's going to be, not just for this course, but for future courses as well. Thank you.